The eye includes several structures in addition to the eyeball. The eyelids, extrinsic eye muscles, lacrimal apparatus, and even the optic nerve are usually included. The orbit forms a protective bony encasement around several of the ocular components. Vision is our most valuable special sense. Loss of the sense of smell or taste, while inconvenient, is not particularly debilitating. Even the loss of hearing comes in as a distant second to the loss of the ability to see. As we study the eye, we will find that the eyeball does not really see anything. What it does is generate impulses that are carried by the optic nerve back into the brain, where the impulses are ultimately interpreted by the occipital lobes of the cerebral hemispheres.
Hi, I'm Mike Shively, and this is Introduction to Anatomy and Physiology. In a previous session, we talked about the autonomic nervous system. Does anyone have any questions about that? Starley.
I was just wondering about hockey players that overdose on Sudafed and what that does to their body.
There are a lot of pharmaceuticals, Starley, that have effects on the autonomic nervous system, and Sudafed is certainly one of those. It's a sympathomimetic drug, which means that if you take that particular pharmaceutical, it stimulates certain parts of the body in the same way that the sympathetic division of the autonomic nervous system does. Now, the clinical purpose of Sudafed, of course, is to dry up secretions in the nasal cavity and so forth and so on, but it has been abused. Certainly some professional hockey players have been accused of abuse of that particular drug, at any rate, because it, remember the sympathetic division is a fight or flight division, and hockey has been known to be a kind of a fight or flight type sport. And as a consequence, to kind of get ready for a game and get themselves fired up even before they get on the ice, some professional players have used that particular drug. Matter of fact, most pharmaceuticals have some kind of an interaction with one division or the other of the autonomic nervous system as perhaps just side effects. Yes, Amber?
I was curious about like the recent ephedra stuff. What does that exactly do to your body, and what harms did they find that took it off the market recently?
Ephedra, coming from actually derived from a plant by that name. I don't know if the versions that are sold are actually from that plant, but there is a plant called ephedra that has that particular same material in it. It's also another sympathomimetic, and I haven't followed that in the news, so I'm not sure exactly what was happening that caused them to take it off the market, but it has similar kinds of effects as the Sudafed. Well, today we want to talk about the eye, and we'll spend a couple of sessions on the eye. Today I primarily want to emphasize eye structure. Now, you know that the eye is closely associated with the nervous system. Matter of fact, we can consider the eyeball to be kind of an extension of the brain, at least in one way of thinking. And its overall purpose, of course, is to generate impulses that are going to be ultimately interpreted as the sense of sight or vision. But when we think of the word eye, I want more to come to your mind than just eyeball, because certainly there are other structures involved. I think on that pre-taped video segment that we just watched, they mentioned extrinsic eye muscles, which move the eyeball around. They mentioned the optic nerve, which actually physically attaches to the backside of the eyeball. And they mentioned the lacrimal apparatus, which actually is involved in producing some moisture to keep the eyeball wet. Why would we need to keep the eyeball wet? What would it matter if the eye dried out? Eva?
Well, it has to move around a lot, and so you kind of need some kind of lubrication to keep it.
Need some lubrication because we do tilt the eyeball in different directions. Is there something beyond the lubrication, Susan?
Well, it's a fragile tissue, I guess you would say, that covers it, and you have to keep it moist to work. Isn't that right?
That's true. As a matter of fact, if the proteins that make up the cornea, which is the clear part that the light has to pass through initially, if those proteins become too dry, they actually undergo some denaturization changes that causes them to become opaque, which certainly wouldn't be conducive to normal vision as we know it. So we have to keep the eyeball wet. Matter of fact, if any of you have been in conditions where your eyes got kind of dry, maybe out in a wind or cold, particularly cold wind, you know, and maybe you noticed you started tearing a bit because of that irritation. They actually make some artificial tears that can be instilled in your eyes because some individuals, the lacrimal gland does not produce sufficient lacrimal secretion to keep the eyeball as wet as it really needs to be. So that's certainly the purpose on keeping the eyeball wet. Bryce?
Is there some other way besides the artificial tears that you could supply fluid to the eyeball if your lacrimal glands don't work?
You know, there have been some experimental surgical techniques. They're not performed very often in humans, but in some individuals where the lacrimal gland is not producing adequate lacrimal secretion, one of the ducts of one of the salivary glands can in fact be translocated up into the conjunctival sac of the eye so that instead of producing saliva, which normally go into the oral cavity to lubricate ingesta during the chewing and swallowing process, that saliva diverted from that particular salivary gland can be used to keep the eyeball moist. And it does work because we have several pairs of salivary glands. The downside, there's a little relearning it has to go on, of course, because every time that you go by a McDonald's, that particular eye waters. So, Eva?
Are the lacrimal and the secretions from those glands in your mouth, do those have the same enzymes in them? Wouldn't that be harmful to your eye to have those digestive enzymes?
Well, one of the things that a lot of people are surprised to find that salivary glands, basically, the secretion is primarily water. There's only one enzyme. It's an enzyme that breaks down starch, a very weak amylase that breaks down starch. And so there's no harm in putting that secretion onto the surface of the eyeball because no parts of the surface of the eyeball are made of starch, so there's no degradation that occurs. It's 99-point-something% water, and so the main component, of course, is the water. All right. I want to, let's take a look then at a graphic. Let's look at this graphic that talks about the overall components of the eye. So when you think of an eye, think eyeball and optic nerve, which attaches to the back of it. Remember, the optic nerve is one of the 12 pairs of cranial nerve. We've got a left optic nerve and a right one. The eyelids, which are skin-covered structures that help distribute the lacrimal secretion over the surface of the eyeball, and also we are able to close those whenever we're in a situation where the eye might be harmed by physical trauma. You see something coming straight at your head, you typically close your eyes and duck. Ocular muscles, which are involved in moving the eyeball around so that we can point it at different objects to see different things. And then, as we've been talking about, the lacrimal apparatus, which is involved in producing a secretion called lacrimal secretion, or out on the street we commonly call that just tears, in order to keep the eyeball wet. So those are the main components of the eyeball, of the eye and the eyeball. When we think of eye, most of those structures are housed within a bony structure called the orbit. And if the camera can slide over here to the skeleton that we have standing here, this orbit and that one cover the back side of the eyeball, the top and bottom sides, and the left and right sides of each eyeball. It's only the front of the eyeball that's left unprotected, where the eyelids have to come down to close things off in case of impending traumatic injury. So the eye is pretty well protected within the bony orbit of the skull. Do any of you remember from your reading, I know it's been a while since we've talked about the skeleton, do you remember the names of the bones that are involved in forming this orbit? There's 3 of them that form quite a bit of it. Again, if the camera could come over here, the top part of the orbit is actually formed by the bone that forms most of the front of the skull. Hint, hint, front. Would that be a frontal bone? Okay. What we commonly call the cheekbone down here, the zygomatic bone, forms the lateral aspect of the orbit and a little bit of the inferior part. The rest of the inferior part and part of the medial part is formed by the maxilla. And then there are 4 other bones back in the back part of the orbit, or a little further back, that form the rest of it. There's a lacrimal bone, sphenoid bone, ethmoid bone, and palatine bone. Let's take a look at a graphic that will put those in front of us in summary fashion. Frontal bone, zygomatic bone, and maxilla form most of the front part of the orbit. And then there's little bits of the sphenoid bone, lacrimal bone, ethmoid bone, and palatine bone that form the rest of the orbit. To understand the structure of the eyeball, I think we all ought to get involved in a little pen and pencil exercise here, and I want us all to make a drawing. Now, the drawing that we're going to make is actually going to be one of a sagittal section. I want the overhead camera to pop on for just a minute and take a look at this model. This is a model of an eyeball, plastic model of an eyeball. We've sliced right down through it in a vertical fashion in a sagittal type plane so that we can see the structures inside the eyeball. Here's the optic nerve coming in from the back, and the cornea would be at this side. So what we're going to do is make that kind of a drawing, and we're going to do it here with the overhead camera on so that we can label some of these parts as we make the drawing. Sitting here in front is the cornea, and then we'll stream back here to the back. We'll say the optic nerve is back here. I'm just going to label that OPTIC, optic nerve. And this part represents cornea. What color is the cornea?
Clear.
Clear. It's transparent, isn't it? So if someone has blue eyes, it's not because their cornea is blue. It's actually a deeper structure. I'm going to give a little thickness to this cornea. And then we'll do the same thing with this part back here. Now, this other part on the outside of the eyeball is called the sclera. So the cornea and the sclera make up the outer parts of the eyeball. Do you remember during the Revolutionary War, the colonists were a little short on ammunition, and during one of the battles, one of the guys in charge of the military said, "Don't shoot until you see the scleras of their eyes." Do you remember that? Maybe you haven't had that history lesson yet. These two things together, the cornea, the transparent part in the front, and the sclera, quote, the white of the eye, make up what's called the fibrous tunic, the outermost layer of the eyeball, because there's a lot of fibrous connective tissue within that. So these two things make up the fibrous tunic. Now, if we draw the deeper structures, we will make a little projection like that, and then one just a little further back, and then come on around like this. We're drawing a deeper layer now of stuff. I realize everything I'm making here is black against a greenish background, but this that we just put on is the next layer down of 3 layers from outside to in, and this layer that we just drew is called the vascular tunic. It's called the vascular tunic. See if we can get this stuck in here. It'll be a little bit tight. So these two things together made the fibrous tunic, the outer part, and then the vascular tunic actually has 3 components. Now, I'm going to label these just 1, 2, and 3. There's number 1. Here's number 2. And then the rest of this back here is number 3. This first part actually is the iris. Number 1 is the iris. So if you want to write number 1, iris. And the hole in the middle of the iris, who knows what that hole is called? Adam?
The pupil?
The pupil. Does the pupil change size?
Yes.
Yes.
It does. And under what conditions will the pupil get larger?
Dark
Darks.
light.
If you're in a relatively low light environment, or if you remember, if you get mad or scared, the pupil will dilate as well. The physiologic purpose of pupillary dilation is to actually allow more light into the eyeball. because the photoreceptors are back in the back part of the eyeball. We haven't even got there yet in our little drawing. But if we're in very low light conditions, a pupil will dilate in order to allow more light in to the photoreceptors. If we're in bright light, if you go outside on a sunny day without hat and sunglasses on, take a look at somebody out in the parking lot on a bright afternoon, what will their pupils be? Very small and constricted down. It turns out that the photoreceptors within the retina have a certain amount of light that is optimal for their impulse production. They convert light into nervous impulses, the rods and cones do. And if the light is too dim, then we need to get more light to them so that they can do their job. If the light is too bright, it tends to overdrive them a bit, and so will constrict the pupil down. Adam?
Why would they dilate during a fight or flight response?
Well, we'll get to this actually when we talk about the smooth muscle structures that change the diameter of the pupil a little later. But the fact of the matter is the pupillary dilator muscle is controlled by sympathetic division. It's smooth muscle tissue, and it's controlled by the sympathetic division of the autonomic nervous system. So if you get mad or scared, then your pupils will dilate as well. Have you ever heard the expression, well, he was so mad he couldn't see straight? Have you heard that? There actually is some physiologic basis for that expression, because under a condition where you're really irritated or scared, the pupil will dilate regardless of what the light levels are. And if the light levels are already pretty bright and the pupil goes ahead and dilates, it'll overdrive the photoreceptors and it'll inhibit normal vision. Amber, do you have a question?
Yeah, I was wondering like how and why when you go to the eye doctor to like have your eyes checked, sometimes they dilate people's pupils and they won't go back for a while.
Okay, the reason that when you go have an eye exam that they put actually some drugs into your eye that will cause the pupil to dilate. What they're trying to do is they want to see back into the deeper part of your eye so they can see what the retina looks like. And so it's a matter of looking through a little tiny knot hole or looking through a great big knot hole. You're going to get a better view of the ball game if you're standing in front of the great big hole in the fence. And so the optometrist or ophthalmologist will actually dilate the pupils in order to be able to have a bigger hole to look through so he can see in there better. Starley?
Sorry, I heard a stat that you can tell if someone's lying by their eye dilating. Do you think that's caused then by the same thing, the fear of maybe when they're lying?
There's some autonomic nervous system involvement in both pupillary constriction and pupillary dilation. Now, I don't know how reliable it is. I'm sure a pathologic liar could look at you straight and not change the diameter of their pupils, but I'm sure there could be something to that. All right, let's go back to the overhead for just a second. So again, number one is the iris, and of course it actually makes a circular ring-like structure, and this hole in the middle of the iris then would be the pupil. Now number two on the drawing, if you'll label that ciliary body, it's C-I-L-I-A-R-Y, ciliary body. And the ciliary body actually suspends the lens. So I'm going to draw the lens in here. We'd have a side view of it, if you will. It's thicker in the middle and thinner on the edges. And the thing that actually doesn't just hover there in space like I've drawn it, but there are actually some little tiny proteinaceous fibers that come off the edge of the lens and attach themselves onto the edge of that ciliary body. Number three, the third part of the vascular tunic, is the choroid. I'll just write it right here. I think I've got room for this one. C-H-O-R-O-I-D. So again, let's repeat those three things. Number one was iris, number two is ciliary body, and number three is choroid. Now the reason these things are all called vascular tunic is because this particular layer of the eyeball has a lot of blood vessels in it. It's well vascularized, so that's where it gets that name. It also goes by a different name. The vascular tunic is also called the uvea. U-V-E-A. And you'll see another variation of this would be uveal tract, vascular tunic, uvea, or uveal tract. All of those are synonymous. All right, the third and deepest of the three basic layers of the eyeball. Remember, this was fibrous layer here, cornea and sclera, then vascular tunic. The third layer of the eyeball, the deepest components of the eyeball, are umbrellaed by the term internal tunic. I'm just going to write that down here. And there's actually three components, and we've already got some of these components drawn up here. The lens is one. I realize this is kind of small. We're trying to jam a lot on this one drawing. Secondly, the chambers of the eyeball. Now, these fluid-filled spaces we haven't talked about yet. Behind the lens is a very large one, and in front of the lens, kind of between the ciliary body and the iris, is a small one. And then in front of the iris, between the iris and cornea, is a small one. All three of these areas are fluid-filled chambers. And starting at the cornea, just behind it, this one right here, is the anterior chamber. So when you think anterior chamber of the eyeball, think this is the area of the eyeball that is between the cornea and the iris. Now, if you go on through the pupillary opening and get into the very small space between the iris and the lens, that is the posterior chamber of the eyeball. So anterior chamber and posterior chamber of the eyeball are continuous with each other through the pupillary opening. Now, the fluid that one finds in the anterior and posterior chambers is called aqueous humor. Aqueous humor. And although it's completely separate from lacrimal secretion, which was on the outside of the eyeball, it too, as you might suspect by its name, is a water-based fluid. It's primarily water. Now, if we go on behind the lens, let's go back to this overhead camera for just a second. Let's go back behind the lens, and this large area right in here is called the vitreous chamber. I don't know if I've got room to write it big enough to see or not, but I'm just going to try. Vitreous chamber. So we've got anterior chamber, posterior chamber, then the lens, and then the vitreous chamber. So the lens actually separates physically by its presence there. It separates the vitreous chamber from the posterior chamber. Now, the fluid that's found here in the vitreous chamber is a gelatinous, transparent fluid called vitreous humor. And although the aqueous humor in these first two chambers is slowly but surely turned over because it's secreted continuously and reabsorbed continuously, the vitreous humor back here in the vitreous chamber is not. It has basically no turnover. It's static for our lifetime. When she was about 3 and he was about 6, our last two children, Jeffy threw a spike nail, just for the fun of it, and it struck our 3-year-old daughter in the eye, and the end of that nail went right through here and poked a hole right through there. Now, I was at a shopping center, and I got a call over the loudspeaker. And, you know, that's a real shocker when you're out in a public place and somebody, you know, you get the call, you hear your name, Mike Shively, please report to the emergency room of the hospital. And I check myself over, I'm okay. And so then you realize, hey, this is not a good news thing. You know, why do they want me to come to the emergency room? I'm a veterinarian. What can I do for somebody there, you know? So I get there, and by that time, our daughter's already in surgery. Basically, they spent about an hour and a half sewing this hole back together. And we spent an hour and a half of misery wondering what was going to happen. The reality is it healed up with little or no scarring. You can look at her eye today and you can't even tell it. An ophthalmologist could look at it and tell, you know, when he looked at it very closely, but she does not have perfect vision, but it has nothing to do with the little scar. Fortunately, it's clear over here to the side. If it had been out in the middle, the scar tissue might have, you know, been in the line of sight and might have interfered somewhat, but it healed up miraculously, and she doesn't have any visual deficit related to that particular incident. Incidentally, I use the term optometrist and ophthalmologist. Does anybody know the difference? Both of them deal with eyes. Adam?
Isn't an optometrist, they just deal with vision correction, where an ophthalmologist actually deals with diseases and deficiencies in the eye?
That's a pretty good summary. And an optometrist deals primarily with visual deficits and their corrections. And an ophthalmologist is actually a physician, gone to med school first, and then specializes in diseases and conditions involving the eye. So basically, optometrists can do fewer things than ophthalmologists can do. On the other hand, as soon as an ophthalmologist figures out he has a visual deficit, he will, in fact, send that person to an optometrist to have them deal with the glasses or contacts or whatever. Any questions about this basic structure now? We had 3 layers. Let's make sure we've got the fibrous tunic on the outside. Matter of fact, let's look at a graphic. It'll summarize the whole thing. I think I've got it on a graphic here. So we'll look at this next graphic. Structure of the eyeball. Fibrous tunic, the outermost layer. 2 major components, cornea and sclera. Vascular tunic, 3 major components. This is the second layer in now. Iris, ciliary body, and choroid. I didn't say too much about the choroid. The choroid kind of lines the rest of the sclera there. We had that little projection that was called the iris, little projection called the ciliary body, and then the rest of that vascular tunic is the choroid. On the internal tunic, we mentioned a couple of these things. I don't think I say anything about the retina when we had our drawing up there, but the lens is one component. The retina, which we're going to talk about a lot more later, is another component, and then we did talk about the 3 chambers. The front 2, anterior and posterior chambers, being filled with aqueous humor, and the back chamber, the one behind the lens, being filled with vitreous humor. Any questions about those basic concepts on structure? We need to go back and talk about some of these individual things quite a bit more, but Starley, do you have a question?
Just in the vitreous chamber, if that ever gets broken or popped, is there any way to refill it?
You know, they may have some artificial material, but when you get an injury deep enough into the eyeball, a traumatic injury, deep enough that there's a loss of vitreous humor, that's a very serious situation. It's silly. Why do we have these fluids in there anyway? I mean, why have any fluid at all? Adam?
So the eye doesn't collapse?
So it doesn't collapse. I mean, we need air in basketballs or they don't work well. We need fluid in eyeballs or they don't work well. We've got to have the thing blown up into its normal, somewhat spherical, but not perfectly spherical configuration in order to allow spacing between certain things. The lens has to actually focus light rays back onto the retina, so it needs to be X amount of distance away from the retina to do that properly. Incidentally, I've got a plastic model of a lens here. If we go to the overhead camera, let's take a look at one. We put one on the drawing. Now, this is a front to back view, and this happens to just be made of plastic. It's just a plastic model from one of our models down in the anatomy lab. And here it is on edge. Does this remind you of the lens in Grandma's magnifying glass? You remember that? This is the one that you used to go out on a sunshiny day and maybe try to turn some leaves on. I don't know if I can't quite get this to magnify because it doesn't show up well enough, but you could actually take this lens and hold it a certain distance away from some dry leaves if you couldn't find the matches in order to what? Start a fire. Or down in Texas, you can take these things and fry ants with them if you get the focal point just exactly right at the right distance. Why would you want to fry ants in Texas? Because everything in Texas bites or stings. I have a personal testimony, having spent 15 years there. It's so nice to be here in Utah where you can actually go out in your yard barefoot and nothing really attacks you. Let's go to the overhead camera and take a look at this model of an eyeball. And this is a section similar to what we drew. Turns out that it's pointed the other direction, but these structures here are the eyelids, okay? This is the cornea, and the space in here filled with aqueous humor would be anterior chamber. This is the iris here. Now, if someone has blue eyes or brown eyes or hazel eyes or whatever color eyes they have, it could be a function of the contacts they have in, of course, but it can also be a function of what color the front of that iris looks to you and I as we look into their eyes. So brown eyes means their iris looks brown to us as we view it. Then this structure is the lens. Now, during its development, the lens actually has a blood supply. So there's some blood vessels that come in from the back of the eyeball here and actually cross through the vitreous and serve as a nutritional source for the developing lens. However, by the time that we reach adulthood, actually well before we reach adulthood, these vessels degenerate. And so the lens itself is just a proteinaceous structure. It has elastic characteristics. It always wants to ball up or become more or less spheroid, but it is held in place again by these fibers. These, I don't think I gave you the name of those. They're called zonular fibers that actually attach to the ciliary body. And we have the ability, we'll talk about this in a future session, but we have the ability to change the thickness of this lens in order to focus images of things that are far away versus images of things that are close up. And if we're going to look at something far away, we need the lens to be relatively thin from front to back, a skinny lens from front to back. If we're looking at something less than 20 feet away, we need the lens to be thicker. And the closer that object is, the thicker the lens has to be. I tell you what, here's something I want you to do right now. You're writing some words there on the paper. I want you to take your notebook or whatever, your notes, and I want you to hold them out at arm's length. Everybody hold them out at arm's length, and I want you to look at those letters or words that you've written. And now bring the thing in slowly towards your face and keep coming in until those letters blur out and you can't keep them from going blurry. Now, some of you are just a very few inches away from your face, and some of you are several inches away from your face. But there will be a point at which you can no longer keep those letters in focus. They will be blurry. Okay? All right, you can change now. Once you're doing that. Now, what was happening as you brought that book in toward your face is that your lens had to get thicker and thicker and thicker in order to bend the light rays enough to bring them to a focal point on the retina. But there was a point at which you got it in so close that you couldn't make your lens any thicker. And that's where the letters started to blur out, and you couldn't keep them sharp and crisp. At any rate, we're going to talk about this some more in future session. Now, I want you to notice how big the vitreous chamber is compared to the anterior and posterior chambers here, which are quite small by comparison. Then way back in here, we haven't talked about it much, we're going to talk about it quite a bit more, is where the retina is located, coats the whole backside of the eyeball on the inside. Actually, there's a part of the retina that comes out on the backside of the ciliary body and the backside of the iris, but there are no photoreceptor cells. There are no rods or cones that are located in these parts of the retina. The rods and cones are all distributed in the posterior parts of the retina. Now, there is a place right in the middle of the back of the retina where the optic nerve comes in. I want to change model. Well, let's keep this model up here, and I'll just move over to this part of it. Right here is where the blood vessels come in, and the optic nerve attaches at that location as well. You can see the blood vessels branching out after they come in this particular area. This is called the disc of the optic nerve. And in this particular part of the retina, there are no photoreceptors. Does anybody know what the street name for this structure is, Adam?
Your blind spot.
Blind spot. You know, I want to do something as a group here that will actually demonstrate that blind spot. Here's what I want you to do. I want you to take a piece of paper there, and I want you to let's go to the overhead to do this so they can see what's happening. Let's put a dot there, and then 2 or 3 inches away from that dot, I want you to put a plus, 2 or 3 inches, okay? So we got a dot and a plus. Now, I want you to stare at the dot, close one eye, stare at the dot, and bring the paper in toward your face, all the while staring at the dot. Now, when you get at a certain distance, depending on how far you put your plus from the dot, you're not going to be able to see the plus anymore. In other words, when you start, you're staring at the dot, but you can still see the plus. Keep staring at the dot, and at a certain distance away, which for me is right in here about 8 inches for me in this particular situation, the plus disappears. Now, granted, don't look over at the plus. Keep staring at the dot. Okay, you with me? Now, let me tell you what's happening. You're staring at the dot, so that's what you're seeing. The plus, in this case, is its image is being projected onto a adjacent part of the retina. If you stare straight at something, that object is being projected onto the very back part of the retina, an area that we call the fovea centralis. But peripheral vision objects are projected onto adjacent parts of the retina. And if you happen to get that projected onto the part of the retina that has the disc of the optic nerve, as you do, if you reach a certain place, bringing that paper in toward your face, you'll in fact put the plus, the image of the plus on the disc of the optic nerve, no photoreceptors, you can't see it. And I don't know if you noticed this when you were doing this or not, but you can see the plus and then you can't see it, then as you come in further, you can see it again, because now the image is being projected onto a different part of the peripheral part of the retina where there are photoreceptors. Does anybody remember what the photoreceptors that are located in the retina happen to be called? Eva?
The cones?
Cones is one. Another one?
Rods.
Rods. We got cones and rods. Does anybody happen to know what cones do for us? We're going to talk about this more in the future, but right now, Eva?
They receive light, they respond to well-lit situations when there's a lot of light and color. They let you see
Okay. Cones generate impulses in moderate to bright light conditions. They give us when the impulses that they generate are interpreted by our brain, we have what we call color vision. All colors, including black and white, can be due to cones. And cones, the visual images that result from the impulses that are generated by the cones we have sharper, crisp images. Rods, on the other hand, the other photoreceptor, works in much lower light conditions, gives us only black and white images, and the images that we get from the impulses that are generated by rods are not as sharp and crisp. Now, most of the retina is a mixture of rods and cones. Turns out that the rods outnumber the cones 12 or 15 to 1. So there's a lot more rods than cones, but there's a scattered mixture of rods and cones throughout most of the retina. However, in the very central part of the back of the retina, there's an area, again, called the fovea centralis, and the photoreceptors that are located there are all cones. Think of it as an area that's 100% cones. So when you stare straight at something, you're projecting the image of that object right onto the fovea centralis, central part of the back of the retina. It's all cones. They do color vision. They give us sharp, clear images. Okay? All right, that's enough on the retina. Let's move on. Oh, do you have a question, Adam?
Just really quick, is it possible to stimulate rods and cones at the same time? What I'm thinking is, say you're in a very dark room, but there's an alarm clock over in the corner with the light flashing. Is it?
As a matter of fact, it is. And there's an area, like, let's just take the sun going down. Okay, we've got a bright, sunshiny afternoon. Only the cones are working because the rods, that amount of light will overdrive the rods and they can't generate impulses. As it gets dark at dusk, as the light levels come down, there's a period of time there where some of the cones are still working and some of the rods are starting to work. If you can see any color at all, you've got functional cones. So out the headlights of your car, when you see a road sign, you can see colors. That's cones that are working. But if you get into very, very dim light conditions, even though you know there may be colors present, all you can see is shapes, and they're kind of perhaps fuzzy shapes as well, then that's rods. Only the rods are working. Now, when you get in absolute darkness, you go to the center of a cave, for example, neither rods nor cones can generate impulses in absolute darkness. So it takes some light to generate an impulse regardless. And we'll talk more about that next time when we talk about the functions of the eye. Let's talk about eye movements a little bit. Now, you know that you can move your eyeballs within their orbits lots of different directions. You can look up, you can look down, you can look to the left, and you can look to the right. And it turns out that there are skeletal muscles, thin, elongated strap-like muscles that attach in the bony orbit and attach to the eyeball, which move this eyeball around. I want to go to the overhead camera for just a minute, and we'll pretend like we've got an eyeball sitting right here. There's a muscle that comes up to the top of the eyeball and goes back to the back of the bony orbit, one that attaches on the bottom of the eyeball and goes back to the back of the bony orbit, and one on each side, right side and left side of each eyeball. These four strap-like muscles are the rectus muscles, one above, one below, one on the medial side, and then one on the lateral side. And so we have four rectus muscles. And let's go to a graphic that names those so that you can see their official names here. Muscles that move the eyeball. The four rectus muscles are superior rectus muscle, inferior rectus muscle, medial rectus muscle, and lateral rectus muscle. And it's pretty obvious that if this one contracts, it's going to rotate your eyeball so that you look what direction? Up. If this one contracts, your eyeball will rotate down, okay? Medial, that eyeball would rotate toward your nose, toward the median plane, yes? And lateral rectus muscle, if it contracts, it'll rotate laterally or out toward your ear. Your visual axis will rotate that direction. So these four rectus muscles can basically move the eyeball in any particular direction. Obviously, if you need to look up as well as to the right, you know, you're going to contract two different ones at the same time and so forth and so on. In addition to these four, there are two muscles that are called oblique muscles. There's superior oblique muscle and inferior oblique muscle that also attach to the eyeball, and they tilt the eyeball just a little bit. I'm not too concerned that you understand how these work, but I think you can logically figure out how these work. Bryce?
So do the oblique muscles make it so you can watch TV on your side?
I don't know. I get confused whenever I lay down on my side. I can't figure out whether they're messed up or whether I'm messed up, but I don't know. So those are muscles that actually move the eyeball around. I'm going to bring a model up here. I want to look at it with the overhead camera. Let me rotate it around this direction just a little bit so you can see the opening here. And this is upper eyelid. And this shows some other structures. But here are the muscles. Now, they've clipped them off on this model, but they would stream on back to the posterior parts of the bony orbit where they would attach. So these are muscles that move the eyeball around. Those are all skeletal muscles. They're voluntary. We can control them at will and move our eyes in the direction that we want to move them. Anatomy students have a little memory aid. I think I might have mentioned this once before when we were talking about cranial nerves, but let's review it again on this graphic. There's a little memory aid that they call LR6, SO4, and the REST 3. This is a memory aid that anatomy students use to remind them that the lateral rectus muscle, that's the LR, that particular muscle is innervated by the 6th cranial nerve. Does anybody remember the name of the 6th cranial nerve? Eva?
The abducens nerve.
Abducens nerve. So the lateral rectus muscle is innervated by the abducens nerve. The superior oblique muscle is innervated by the 4th cranial nerve, and that happens to be the trochlear nerve. And then the rest of the eye muscles, including the smooth muscle structures inside the eyeball, are innervated by the third cranial nerve, which is the oculomotor nerve. LR6, SO4, and the rest 3 just reminds anatomy students which eye muscle is innervated by which cranial nerve. Starley.
Sorry, when people have a lazy eye, is it a weak muscle?
I don't know if you've thought about this much, but if you look at something that's fairly far away, the visual axes of your two eyes are more or less parallel, are they not, in reaching that object? But the closer an object gets to your face, the more your visual axes have to converge. In other words, if you take your thumb and hold your hand out here at arm length, okay? Now pull your thumb in toward your face and keep looking at it with both eyes. And as I'm looking at you, or if, I don't know if the camera can see me or not, but I am now pretty cross-eyed, am I not? Because why? Because in order to get both eyes to look at that same object at the same time, I have to bring the visual axes in. We have to go, quote, cross-eyed, unquote. It's called strabismus when the visual axes aren't lined up properly. Now, if you're looking at the object and you're focusing both eyes on it and everything's working okay, that's not lazy eye or cross-eyed or anything. But if one eye is going this direction and you want the other eye to go this direction and it's off over here someplace in Nebraska as opposed to where it's supposed to be, then we have a condition of strabismus. Strabismus, it's called. If the visual axes are going too wide or going away from each other too much, we call that divergent strabismus, and that's the lazy eye is kind of the common name for that. On the other hand, if the visual axes are pointed in further than they need to be, and we have a cross-eyed situation, that's called convergent strabismus. So strabismus when the visual axes aren't properly aligned, divergent if they're going away from each other too much, and convergent if they're coming toward each other too much. Susan?
Why is it that we can go cross-eyed but can't look out?
There are, in other words, I can go cross-eyed anytime I want,
Right.
but I can't stand here and make this eye go off to this side. There are a few people that have that talent, and I never did develop it myself. But there are some individuals. And of course, there are some corrective surgical techniques to help people who have visual axes that tend to be lined up wrong. They can go in and actually do some surgery on the extrinsic eye muscles. And if we have convergent strabismus or cross-eyed situation, they can either loosen up the medial rectus muscles or tighten up, if you will, the lateral rectus muscles in order to get these visual axes to come back out a little bit.
How do they bring that out in order to do the surgery if all the muscles are, they're attached in the back?
Well, what they'll do, of course, this is not a local anesthetic thing, Starley. They're going to put you clear out, and then they can proctose the eyeball. They can actually, when you're anesthetized, they can actually pull it out of the bony socket a little bit and get to the eye muscles to do these techniques. Amber?
I went to the eye doctor, and he told me that, like, my left eye is so bad that when I see, it doesn't like my brain doesn't like what it sees, so my left eye basically doesn't really work. It uses mostly my right eye. What is that called?
Well, we all have an eye that is the, quote, dominant eye in us. And let's do the thumb thing again. I want you to hold your thumb up, and I want you to line it up on me. Keep both eyes open. Line your thumb up on me. Now, if you look through your thumb at my face, if you got both eyes open, you notice you see two thumbs? Okay. Now, if you look at your thumb and see me in the background, how many Mike Shiveleys do you see? Two. That's scary, isn't it? Now, now let's do this. I want you to line one of those thumbs up on my face. Okay? Now, the other one is not lined up on my face. If you close an eye and the thumb is still lined up, then that's your dominant eye. The eye that you close, that you left open, is your dominant eye. Because you have a tendency to line things up in these kinds of situations with your dominant eye. Okay, you can put them down now. At any rate, going back to your question, Amber, we all have that to some degree. In some individuals, in perhaps your case, from what you say, your dominant eye is so much more dominant than your other one that the other one tends not to be involved hardly at all. Some people have a visual deficit in one eye and don't have a visual deficit in the other eye, and they'll tend to use the eye that gives them good, clear vision in, and so that will become their dominant eye, and they'll tend not to use the other one. I have a little bit of a visual deficit in one eye. It's worse than the other one, and so I tend to use my right eye is better than my left eye, and I tend to, whenever I'm trying to read really small print, I just leave the left eye out of the program for a little bit. So.
Does that have a name or anything?
I'm sure there is a name. You know, ophthalmologists and optometrists have a name for everything. I don't know what it is for, you know, a clinical condition where one eye tends to be exceptionally dominant.
Is there any way to like fix it if it's really?
Depends on what's causing the situation. Is it some kind of visual deficit that glasses or contacts could correct for that, quote, weaker eye, or the one that's left out? I don't know. But some of the situations they can probably do something, and others probably not. Oh, a couple other things I want to cover before we finish today. Let's go back to the overhead camera for just a second, and let's draw our eyeball shape here again one more time. Now, we haven't talked about eyelids, and I want to bring an eyelid down. We'll put some eyelashes on here. Let's see. With Maybelline, they might come out to about here. All right, then we'll put a lower eyelid on. Now, in addition to being able to close these lids in order to protect the delicate structures of the eye, we mentioned the fact that they distribute that moisture, the lacrimal secretion, over the surface of the eye. If we actually take a look at the layers, on the outside here, we're going to have skin. And deep to that, there's actually a little thin skeletal muscle called the orbicularis oculi muscle. And then finally, on the lining in here, there's a mucous membrane called conjunctiva. Let me write that word down. Conjunctiva. So we got skin on the outside, orbicularis oculi, and conjunctiva. And there's some mucus-secreting cells in the conjunctiva that are busy secreting a mucus-like material. If your eye gets irritated, they go into overtime. Have you ever been out in real dusty conditions and then you went to sleep that night and the next morning you tried to open your eyes and your eyelids were kind of glued together with stuff? You ever had that experience? Mm-hmm. What's happened in that situation is the irritation has caused the mucus-secreting cells within the conjunctiva to go into overdrive. They've secreted a lot of this mucus, and when that mucus dries, it kind of glues things together. So that's what do you call that in the morning you wake up? You got those little things in there. What do you call those? Eye sand? That's just, yeah, eye boogers. I won't say that on video, but nonetheless, it's dried mucus is what's causing that. Now, I want to change my drawing here just a little bit because I want a little more space between the eyeball and the eyelid. I want to show you something else. So I want to make the eyelid up here a little further away. We'll just put the upper lid on. Those of you who wear contacts know that the radius of curvature here on the cornea allows the contact lens to more or less sit right there, except when you run into somebody's elbow or get bumped in a certain way, this contact lens can get dislodged, can it not? And it can go all kinds of different directions, but it can go maybe up in here. You never, however, have to worry about it going all the way around here behind the eyeball and clipping off the optic nerve, which would be inconvenient. and yeah, the reason for that is the conjunctiva lines the inner surface of the eyelid, then it comes kind of reverses direction and comes down here on the eyeball. So there's actually, it's kind of like the bottom of a hip pocket on blue jeans. There's only so far you can go down in that hip pocket with your billfold or your hand, and there's only so far back this direction that that contact lens can displace. Okay? All right, one more thing I want to show you and talk about today, and that's the lacrimal apparatus. And I want to bring a model back up here for the overhead camera. And we'll take a look at this model on overhead. The lacrimal gland is about the size of a dime, and it's located on the upper lateral aspect of the eyeball. It's flattened out, very thin, like a dime is, and its job is to produce lacrimal secretion, or tears. Those tears, or that lacrimal secretion, enters that conjunctival space that I just drew through some little tiny ducts, and you can see these little white structures right here. So basically that secretion is released freely onto the surface of the eyeball, deep to the eyelid. The eyelids then, by opening and closing, distribute that lacrimal secretion over the eyeball, and then the excess lacrimal secretion drains out through two little tiny openings, here and here. I think you can see these little black dots, perhaps on camera. And those little black dots, if I point them out maybe with this pointer, you can see here's one here on the lower eyelid and one here on the upper eyelid. Each of those openings is called a lacrimal punctum, P-U-N-C-T-U-M, punctum. Leading away from those openings are little tiny channels called lacrimal canaliculi that carry the lacrimal secretion back here into a dilated area called the lacrimal sac, S-A-C, lacrimal sac. So let's review that again real quick. Lacrimal gland, the ducts of the lacrimal gland, the secretion's flowing freely across the cornea and sclera, goes into a lacrimal punctum, drains into a lacrimal canaliculus, is what these channels are called, then into the lacrimal sac. From here, the secretion drains down through a nasolacrimal duct into the nasal cavity, where the excess lacrimal secretion is dried by evaporation. Except when you're emotionally disturbed and the lacrimal gland goes into real high gear, produces too much lacrimal secretion, then your nose will run because the evaporation processes can't dry up that degree of secretion. Or perhaps if you get something in your eye or you're peeling an onion, you know, you can get an irritation effect. The lacrimal gland goes into high gear and your nose will run. Or perhaps you'll get so much lacrimal secretion that the excess will spill out over your lower eyelid, and we'll look at you and say, "Why are you crying? The test is not until next week." Okay? Anybody have a question? All right, they're rolling credits, so I think we've finished.